Okay, very good morning, my dear friends. Uh, in the regarding of CPGET 2023 free online coaching for botany and plant sciences. Today's one of the most important area for the all competitive exams in plant sciences, that is genetics. So plant genetics. So one of the eminent resource person, expert in genetics, and a huge experience personality, eminent botanist, becoming Dr. Bhageerath sir. Sir has working as an assistant professor, Department of Botany, GDC, Rangasaipet, Warangal district. Sir has huge experience in the field of plant genetics, especially the specialization. So I would like to invite Dr. Bhageerath sir. He is my beloved brother. Please welcome, sir, and share your screen. Start your session, sir. Please welcome.
Thank you, sir.
Right.
My voice is audible?
Yes, voice is clearly audible, sir. Nice to meet you. Please welcome.
Yes, sir. So, okay. Thank you, sir, for giving this wonderful opportunity to share my knowledge with our stakeholders, the important stakeholders, our UG students they are studied in various degree colleges.
Yes, sir.
Today, I will explain about some important topics in genetics area.
Yes.
Sir.
You please click on slideshow. It is very clearly visible, if possible. Slideshow.
English medium or Urdu medium करते सर कॉल से.
Is it clear?
काफी के.
Okay, sir. Now.
Okay. Okay, sir. Please proceed.
Okay. So, genetics is one of the important and interested field in the entire field of the biology. So, one of the branch of biology which deals with in the previous 1800 or 1900 earlier years, we discuss about in the genetics, you only discuss about the variations or mutations and heredity, but later on some scientific theories and experiments which reveals about the true secrecy of the heredity, the important chemical present in the nucleus, that is the DNA. That's why the genetics branch which deals about three important parts, that is structure of gene, as well as synthesis of gene and function of gene. So not only these three aspects, we can discuss about how the parental characters, how the parental characters are transmitted from one generation to another generation. So what is the mechanism? What is the secrecy in the transmission of the characteristics? And as well as once we can observe in a particular generation, we can see a different characters which are not related to the parents. So that is kind as mutations. So genetics is one of the branch which deals with structure of gene, synthesis of gene, function of gene, and also heredity and mutations also. So before going to the genetics area, we can first the chronological history of the genetics. Before in the 1800s period and up to 2000. So what are the changes? What are the different theories and what are the new information revealed by different scientists by their scientific experiments? So genetics is the study of genes, heredity, and genetic variations in living organisms. So first time, the genes, the term coined by William Johansen in 1909 is one of the important bit frequently asked in different competitive examinations like PG entrances and set examinations like. So a heredity factor that constitute a single unit of heredity material in correspond to a segment of DNA that code for synthesis of a single polypeptide chain. Gene is nothing but it is a tiny or the smallest part of the DNA which contribute a particular character, or it helps in the manifestation of the particular character, or it will be useful to synthesize a particular product like enzyme or protein or any other important metabolite in the living cell. So heredity is known very well, the passing of phenotypic characters from parents to their offsprings through their asexual reproduction or means of sexual reproduction. Is it clear? Hello? Genetic variation. Genetic variations is nothing but so differences or dissimilarities between the individuals in a group of populations. So we can call it as genetic variations or sports. First time the genetics, the term proposed by English biologist William Bateson in 1909. Gene అనేటువంటి term first time coin చేసిందేమో William Johansen. Genetics, this word coined by William Bateson in 1909. So genetics is relevant to all fields of biological disciplines, including medicine, agriculture, anthropology, biochemistry, microbiology, biotechnology, ecology, and physiology, as well as psychology. So broadly, this genetics branch divided into two areas. One is classical genetics, another one is modern genetics. In the classical genetics, we can deal with only how the characters or traits are passing from one generation to another generation. Any differences are found in a particular generation, we can coin as it as mutation. So this is explaining in classical genetics period. So later on in the modern genetics, which is divided into so many categories, so many types, like biochemical genetics, population genetics, and microbial genetics, plant genetics, animal genetics, like this. So this is the modern genetics. We can classify it according to our feasibility, what are the research going on in the plants. Can you give anybody example different plants and animals or microbes are utilized for the tools for the genetical study? Hello? Anybody give examples for plants, animals, or microbes? So scientists are taken as experimental tools for their scientific study. Can you give any example? Hello? Hello? My voice is audible. Hello? Hello? You know Mendel. What is the experimental tool for the Mendel experiment?
Pea plant, sir. Good morning, sir. Pea plant.
Pea plant, Pisum sativum. So you know very well that is Drosophila melanogaster. That is fruit fly. It is a good example for genetical study. Yes or no? You are studied in the zoology part or the botany in the genetics part. And one more example is there guinea pigs, rats, Neurospora crassa. These are the famous examples which are selected as a experimental tools. Even Escherichia coli is a good example for the bacteria. So many experiments are conducted on the Escherichia coli. That Neurospora crassa is fungi, bacteria is Escherichia coli, plant is the Pisum sativum, animal is a good example is the guinea pig or rats, Drosophila melanogaster, and the Neurospora crassa, it is fungi. So many examples are given. We are studied in different chapters under whether it is in different topics like crossing over or the biochemical genetics or in any other aspects. So in 1859, Charles Darwin proposed the theory, the origin of species. In this book, he explained about the evolution by natural selection. In 1866, Gregor Mendel published the experiments The monohybrid and dihybrid experiments was conducted by the Gregor Mendel, which was published in Plant Hybridization book, which is the basic theory of the genetics. It is widely ignored until 1900. After 1900, so many different theories are explained. The Mendel laws are wrong in all aspects. Mendel hereditary laws are really, it is applicable to the pea plant and some other organisms, but later on, which is not applicable to all microorganisms or all living organisms. So that's why the experiments was published in Plant Hybridization book by Gregor Johann Mendel. And Frederick Mischer, that is isolates the nucleic acid. Waldeyer coined the term chromosomes. So these are the relevant to the before 19th. In the events in 20th century, in the 1900, the rediscovery of Mendelian laws. Rediscovery of Mendelian laws done by three scientists, Correns, Hugo de Vries, and Erwin von Schermach. So these three persons are again rediscover the Mendelian inheritance laws in 1900. So in 1800s period, in the 1850 to 1866 period, the Mendel laws are discovered, and these laws again discovered in 1900 by three persons named as Carl Correns, Hugo de Vries, and Erich von Schermach. So these three scientist names are also very important in the point of examination. So Hugo de Vries coined the term mutation. Garrad is discovered the alkaptonuria. It is a biochemical genetics. We can explaining about the different type of diseases, alkaptonuria, phenylkaptonuria, human disease as genetic basis. The term genetics was first proposed by the English biologist William Bateson, and a gene was introduced. The term was Johansson. So these are all things we can explain in later chapters also. So we can go for, before start the genetics chapter, we know the some terminologies, common terminologies in genetics. So we can understand genetics chapters before knowing about few terminology, which are very important in the aspect of genetics. Is it clear? What is meant by trait? Trait or character. So it is a distinguished quality of characteristics, typically one belonging to a person or a plant or animal. In science, the word trait refers to a characteristic that is caused by gene. Trait and it is a character. So the external appearance or the phenotypic appearance of the particular animal or plant, we can say in the form of traits. Trait is nothing but is a character. It is a distinguished quality of the characteristics, whether it is height, color, or size, in any different, etc., etc. So next, what is meant by allele? Alleles are very important. Allele are nothing but a pair of genes which are located on the same locus of homologous chromosomes. Allele is nothing but a pair of genes which are occupied on same locus on homologous chromosomes. Allelic genes are sometimes are different, sometimes are same. For example, you studied in the genetics, the tall character in the pea plant, which is determined by a pair of genes, that is capital T, or capital T, small t. Are you followed? Capital T is present. What is that condition is? Homozygous. Homozygous. So we can see in the F1 hybrid, the genes, the allelic genes are different, capital T and small t. That condition is known as heterozygous. What is that? Heterozygous. So heterozygous, which containing two different allelic genes. Homozygous condition, a pair of genes are like, same genes are present in the allele and which are occupied on the same locus of homologous chromosomes. So dominant character and a recessive character. What is meant by dominant character? In some cases, in the heterozygous condition, one gene dominant over other gene. One gene express its dominance over the other gene. For example, tall and dwarf, both characters are expressed by the respective genes like capital T and small t. If capital T is present in the heterozygous condition, which shows the dominance nature over the small t gene. So that is dominant character. So dominant character gene we can express in the capital letter. Recessive gene letter is expressed in the small. Is it clear? So what is meant by trait? What is meant by dominant character and recessive character? In the F1 generation, one of the parental character is mask, that is called as a recessive character. One of the parental character is only expressed in the F1 generation, that is the dominant character. How can you identify the dominant character? In the when we plotting the genes on the chromosomes, the T, capital T, the capital letter indicates the dominance, and the small letter indicates what? Recessive. Are you followed? Hello? Hello? What is meant by
Yes, sir.
what is meant by phenotype and genotype? Phenotype is nothing but it is the external appearance of the trait or character. So that is nothing but is called phenotype. The phenotypic character is denoted by a pair of genes or a genetical composition or genetical constituents, that is capital T, small t, capital Y, small y. So that is nothing but is called genotype. Genotype is nothing but the character which is denoted by a pair of genes. A pair of genes that is nothing but is called a genetical composition. But actually on the chromosome, there is no capital T letter or capital Y. It is a gene only. We can denote it by a letter, whether it is maybe ABC like that, or maybe the character is tall, we can denote it with T only. So like that. So these are the some important terminology before going to the explaining about the genetics. What is meant by trait? What is meant by locus? Locus is the exact position of the gene on the chromosome is called as locus. Loci or locus, plural and singular words we can explain in this. What is meant by locus? What is meant by dominant character? What is meant by recessive character? Allele, homozygous, heterozygous. If a allele is in heterozygous condition, it produces two different gametes. It produces two different gametes. If allele is in homozygous condition, it produces similar gametes. It produces similar gametes. Are you followed? Homozygosity, heterozygosity, or homozygous condition or heterozygous condition. Both conditions are seen in the allelic form. Allelomorph, allele, both are same. Okay. Are you followed? Are you followed?
Yes, sir.
Okay. So Gregor Johann Mendel is basically is a monk or a priest. Is popularized as a father of genetics. Why Mendel is popularized as father of genetics? What is the main reason? Why Mendel is popularized as father of genetics? First time he reveals about the secrecy of the heredity, how the parental characters are transmitted to offsprings, how the traits or characters are passing from one generation to another generation. He tried to explain the secrecy of the heredity without knowing about the chromosome, without knowing about the gene, without knowing about the nucleus. He don't know about the nucleus, he don't know about the chromosome, he never go to the laboratory. Just he observed the surrounding plants. By observing the nature, suddenly he watched a different color flowers are appeared in a particular plant. So that's why he got an idea to conducting hybridization experiments by choosing one of the good plant as an experimental tool, that is pea plant, garden pea plant. What is the experimental tool for his experiments? Pea plant. The name of the scientific name of the pea plant is Pisum sativum. It belongs to which family? Which family? Hello? Fabaceae family. What is that? Fabaceae family. So first time he discovered the inheritance laws. That's why he is popularized as father of genetics. So he conducted hybridization experiments on pea plant. Why he choose pea plant? Why he is selecting pea plant as an experimental tool? Because it is easy to grow or cultivate in the field, and it is an annual plant, and naturally the flowers of pea plant is self-pollinated. We can done artificial cross-pollination whenever required, and the progeny produced in many number, and crystal clarity in the features or traits of the progeny. How many pairs of characters he choose for his experiments?
7 pairs.
Yes, 7 pairs of characters he selected in the pea plant. The pea plant having only 7 pairs of characters only? No, many characters are there, but Mendel luckily choose how many pairs of characters? 7 pairs of characters. If he selected more than 7 pairs of character, maybe he may not get the 9:3:3:1 ratio or 3:1 ratio always. That's why Mendel is a fortune person. అంటే మెండల్ని చాలా మంది ఏమనుకుంటారు, అదృష్టవంతుడు అనుకుంటారు. ఎందుకు అంటే he selected pea plant, among the characteristics he select only 7 pairs of characters. What is the chromosome number in the pea plant? What is the diploid chromosomal number? Pea plant. 2x or 2n is equal to 14. How many? 14. 14 chromosomes are present in the pea plant. So that's why 2n is equal to 7 pairs. 7 pairs of the character. They were easily available. They reproduce quickly. In the short period of time, he observed n number of generations. N number of generations. And the progeny number is also more, because we can cut the one fruit, we got many number of seeds are there. Okay. This shows obvious difference in the traits and a crystal clarity in the expression of the characters. There is no doubt in the expression of the particular trait. Red color means red only. White color means white only. So tall means it the stem height grown up to 10 feet. If it is a dwarf, it is only 2 feet or 3 feet. A crystal clarity between the traits among he selected. So but he don't know about the gene term also. That's why Mendel coined that a character is expressed by the factor only. According to the Mendel, the gene is called as what? Mendel prakaram జన్యు అంటే ఏంటి? Factor. What is that? Factor. Gene అని ఎవరు పిలిచారు మరి? First time the gene term coined by William Johanson. William Johanson. Are you followed?
Yes, sir.
Are you able to understood? అర్థమవుతుందా?
Yes, sir.
Okay. So Mendel's experiments with pea plant, the scientific name is Pisum sativum, L for Linnaeus. Gregor Mendel was one of the first person apply an experimental approach to the question of inheritance. For 7 years, Mendel breeding the pea plant and recorded inheritance patterns in the offsprings. So he was noticed how many characters are present in the offsprings. He selected 2 plants, and what are the characters observed in the parental group? The same characters are present in the offsprings or not. He noticed and recorded in the book. So based on the result, he concluded a ratio. He concluded a mathematical ratio. Based on these mathematical ratios, he proposed inheritance laws. Based on mathematical ratios, he proposed the inheritance laws. దేన్ని ఆధారంగా ఇతడు ఇన్హెరిటెన్స్ లాస్ ను ఫస్ట్ టైం డిస్కవర్ చేశాడు, ప్రతిపాదించాడంటే ఓన్లీ మ్యాథమెటికల్ రేషియోస్. బేస్డ్ ఆన్ ద మ్యాథమెటికల్ రేషియోస్ he proposed the inheritance laws, and he explained the how the what is the mechanism in the maintaining the characters or expressing the characters from parents to offsprings. Characters of parents pass on to their offspring, separate and distinct factors. Today the factors are called as genes that are responsible for inherited traits. So in the hybridization experiments, you know very well, the first step is in the hybridization of the plants is selection of parental plants. What is that? First, selection of parental plants. P antam. Manam genetics రాసేటప్పుడు P means parental or parental plants. You can choose 2 plants as parental. One is considered as male plant, another one is considered as female plant. Okay. So from male plant, we can collect the pollen grains, and the pollen grains are transferred to the second parental plant, that is female plant. This is to prevent the self-fertilization. Mendel cut off the immature stamens structures that produce pollens. In the female plant, we can cut the stamens. That is nothing but is called emasculation. And sometimes we can do bagging also. In the hybridization experiments, we can follow some steps: selection of the parental plants, emasculation, bagging, artificial cross-pollination, and identify the progeny, and study about the progenies F1, F2, F3, like that. So this is the mechanism of the hybridization experiments. So P generation, by crossing 2 parental plants, we can obtain hybrid plants, that is F1 generation. By the selfing of F1 plants, we can obtain F2 generation. So in the Mendel hybridization experiments, we can write P generation, F1 generation, F2 generation. P generation is parental plants. Always the parental plants are homozygous and having 2 different traits, a pair of contrasting pairs. And by crossing the 2 parental plants, we obtain the F1 plants. We can identify the nature of F1 plants, and self-pollination between the F1 plants, we can obtain the F2 generation. Is it clear? So, and moreover, you can remember the 7 pairs of characters is also very important. How many pairs? 7 pairs of the characters. What are the 7 pairs? Flower color, flower position, seed color, seed shape, pod color, stem length. Stem length is tall and dwarf. Pod color is green and yellow. Pod shape is inverted and constricted. Seed shape is round and wrinkled. Seed color is yellow and green. And flower position is axial and terminal. And flower color is purple and white. So these are the 7 pairs of the contrast characters are selected in the pea plant, and conducted hybridization experiments. Mendel was mainly conducted 3 type of hybridization experiments. One is monohybrid, dihybrid, trihybrid also. Somebody is. Please mute yourself. Redmi, ఎవరమ్మా? Redmi, please mute yourself. Redmi phone ఎవరిదమ్మా? Creates disturbance. Hello? Hello? Please mute yourself. Hello? Redmi ఎవరమ్మా phone? Please mute yourself. Hello? Monohybrid experiments. So in the monohybrid experiments, what is meant by monohybrid experiment? Anybody? Hello? What is meant by monohybrid experiments?
Sir, only one character is taken in this experiment.
The hybridization carried out in between two individuals which differ in set of characters. A cross made between the two parental plants which have different set of characters. So for example, we can go for fruit color. Two parental plants are differ in fruit color. For example, height of the stem. The height of the stem is differ in two parental plants. You select a pair of contrast characters. One pair of contrast characters among 7. Mendel choose out of 7. You select one pair that is comes under monohybrid experiments. Is it clear?
Yes, sir.
Okay. So if you select one pair, so the first you write the parental characters. So okay, parental plant, that is you can select a capital T cross with okay. The capital T cross with small t, small t. It's not comes like this. Okay, I will go through one more whiteboard. Okay, we can go for Mendel monohybrid experiments. Before going to that, okay. In the monohybrid experiments, actually, based on the monohybrid experiments, he revealed two inheritance laws. What are the two inheritance laws proposed by monohybrid experiments? One is law of dominance, another one is law of segregation. What is that? Law of dominance, another one is law of segregation. It is a cross between two pure organisms or plant in order to study the inheritance of a single pair of allele. Single pair of allele means when you see capital T cross with small t, small t. So pair means two. One is capital T, another allele is small t, small t. So it produces a phenotypic monohybrid ratio 3:1 in F2 generation, and it produced genotypic ratio is 1:2:1. Are you followed? For example, I ask one question. Mendel, he selected 7 pairs of character. Every time he take a pair of character, conducted monohybrid cross, and always he obtained ratio in the F2 generation. And what is the F2 generation phenotypic ratio as well as genotypic ratio? What is the monohybrid phenotypic ratio and genotypic ratio in the F2 generation? Tell me.
So phenotypic ratio will be 3:1 and genotypic ratio 1:2:1.
It is always similar. Any single pair of characters he selected. And for example, height of the stem is selected. You get always 3:1, 1:2:1. Once again, he selected pod color. And another time he selected position of the flower. So every time he got, what is the ratio? 3:1 and 1:2:1. What is the reason for that? He getting same ratio every time? What is the reason? Anybody know? You don't know? Hello? He always getting same ratio 3:1 and 1:2:1. F1 100% hybrid. So first you observe that in the F1 generation, only one of the parental character is appeared, expressed. Another parental character is masked. Are you getting my point? So for example, seed shape is round is there, wrinkled is there. In the F1 generation, only round is present, wrinkled is absent. Wrinkle is completely absent. But the representative gene is present in the F1 hybrid. Are you followed? I will give when for example, capital T, small t. Are you followed? This is the F1. What is that? Genotype. F1 genotypes are capital T, small t. But here small t is there, but it is unable to express its character. Why? Because due to the presence of the capital T, it shows dominance over the small t gene. That is called as law of dominance. Law of dominance. In any hybridization experiment, whether it is dihybrid, trihybrid, or monohybrid, if trihybrid, 3 characters are expressed. Remaining 3 characters are masked. In dihybrid, 2 characters are expressed. What is that? Round and yellow only expressed. Green and wrinkled are masked. So, what are the 2 genes which are denoted by capital letter, which are coined as dominant gene. What are the genes which are written in small, which are expressed, which are comes under recessive genes? So the dominant gene always dominate the over the recessive gene. So that's why in the F1 generation, only one of the parental character appeared. That character decided as dominant. That gene is called as dominant genes. That particular law is called as law of dominance. Is it clear?
Yes, sir.
Okay. So during the meiosis process, at the time of gametogenesis, the allelic genes are separated.
Sir, your PPT is not visible, sir.
Not visible. Okay, I will go through that. Now,
Yes, sir. Now it is visible.
okay, I will explain. Okay. So the F1 plants always hybrid. Hybrid means the F1 plants are always hybrid, 100% show only one of the parental character. That character decided as dominant character. The particular gene is called dominant gene. So beside the dominant gene, recessive gene is present. But the recessive gene, whenever the absence of the dominant gene, it expresses its total purity of the character. So that's why in the F2 generation, we can see dwarf character. We can see the dwarf character in the F2 generation. Without small t gene in the F1 generation, we are unable to get the character. So that's why Mendel says, according to the monohybrid experiments, two laws are proposed. One is law of dominance, another one is law of segregation. In the law of segregation means the two allelic genes are separated at the time of gametogenesis. They randomly united at the time of fertilization. Again, we get the different combinations. Law of segregation. At the time of F1 hybrid, these two genes are never lose its character, or never blend its character, never change its character, never its loss of its originality. Capital T is there, small t is there, but small t never lose its character. It unable to express its character due to presence of the dominant gene. That is, you have to understood first here. That is called as law of dominance. Is it clear?
Yes, sir.
And in dihybrid or monohybrid, Mendel got same ratio due to these 7 pairs of genes are present on different homologous chromosomes. Different homologous chromosomes. He don't know about the gene's position to the Mendel. That is the actual secrecy of the Mendelian genetics. మెండల్ ఎందుకు పాపులర్ అయ్యాడు? మెండల్ అనువంశిక సూత్రాలను ప్రతిపాదించడంలో ఎందుకు సఫలీకృతుడయ్యాడు అంటే ద 7 pairs of genes which are present on different chromosomes. That's why Mendelian genes are non-linked genes. What is that? Non-linked genes. Mendelian laws are applicable to A, nucleus, B, plastids, C, mitochondria, D, plasmid. Which is the correct answer? Mendelian laws are applicable to A, nucleus, B, plastids, C, mitochondria, D, plasmids. What is the correct answer? Nucleus, because in the nucleus chromosomes are there, 7 pairs of chromosomes are there, 7 pairs of genes are present on different chromosomes, not in a single chromosome. ఒకవేళ క్యాపిటల్ టీ క్యాపిటల్ వై కనుక ఒకటే క్రోమోజోమ్ మీద ఉంటే మెండల్ కి రేషియో వచ్చేది కాదు. ఇఫ్ యు సస్పెక్ట్ ద క్యాపిటల్ టీ అండ్ క్యాపిటల్ వై జీన్ present on the same chromosome, definitely Mendel also got different ratio. Mendel every time get 9:3:3:1 in dihybrid, 3:1 in monohybrid. What is the secrecy means? The 7 pairs of genes which are present on single chromosome or different chromosomes?
Different chromosome.
Yes, that is the secrecy for the victory of heredity loss. Ante Mendel enduku safalíకృత Mendel is a fortune person as per as his experimental studies. Mendel కూడా తెలియదు కానీ Mendel ఎప్పటికూ కూడా 3:1 రేషియోనే వచ్చింది, 9:3:1 రేషియోనే వచ్చింది every time, because the 7 pairs of genes are present on different chromosome. That's why Mendelian loss or Mendelian genes are non-linked genes. What is that? Non-linked genes. In the crossing over chapter, what you are studying about that? Linked genes. But here Mendelian genes are non-linked genes. The law of dominance is you very well understood, and the law of segregation or దీన్ని మనం పృథక్కరణ సిద్ధాంతం అంటాం తెలుగులో అయితే. Law of segregation, the two allelic genes are separated at the time of gametogenesis. Every gamete receive only one factor. Every gamete receive how many factors?
One factor.
One factor, either capital T or small t. ఏదన్నా ఒకటే వస్తుంది గ్యామెట్ లోకి, రెండు వెళ్ళవు. A gamete having only one gene or one factor of allele. That's all. That is a universal truth. Mendel laws is applicable to diploid sexual reproducing organisms only. The Mendel laws are applicable to diploid sexual reproductive organisms. So that is the pea plant. Among the 7 pairs of characters, you always getting this ratio. Chudandi ikkada 2.84:1, 2.82:1, 2.95:1. Almost all it is similar to 3:1. So that is the major thing you can observe here. So this is the law of segregation. So law of segregation of theory. This is the Mendel first monohybrid experiments reveals about the law of dominance and the law of segregation. And the dihybrid ratio, you can choose 2 pairs of contrast characters and made a hybridization or made a crossing between the 2 parental plants which are different 2 pairs of characters. That is comes under dihybrid ratio. How many plants are, how many progeny produced in the monohybrid experiment? 4 plants. How many progeny produced in the dihybrid experiments?
16.
16. How many progeny produced in the trihybrid?
64. 64.
Yes, exactly correct. 4, 16, 64. So, and what are the possible gametes you write from the genotype? So, for example, I will explain. This is the genotype, no? So, CRCW is the genotype here. What are the possible gametes produced from the given genotype? That is, you use 2 to the power of n formulae. 2 to the power of n formula. N is number of alleles. Number of alleles. So, for example, I will write capital T, small t, and capital L, small l, small l, capital B, capital B, small b. What are the possible gametes we are obtained from this genotype? What are the possible gametes obtained from this genotype? This genotype is capital T, small t, capital L, small l, capital B, small b. So how many alleles are there? How many? 3 alleles are there. So what is the formula? 2 to the power of n. 2 to the power of n is equal to 2 cube. What is that? 2 cube. 2 into 2 into 2. How much it is? How many gametes? How many gametes?
8.
8 gametes are produced from this genotype. So this is a simple formula applicable to whenever you write. For example, as per Mendel's dihybrid, what you write? Capital Y, small y. Okay. Capital R, Small r. How many gametes you are produced from this genotype?
4.
4 gametes. So for example, it is homozygous condition. How many gametes are formed? So homozygous condition means capital Y, capital Y, capital R,
Capital R.
capital R. How many gametes?
4.
4 gametes, but all are same. All are
Same.
same. So that's why we write only one here in the dihybrid experiments, capital Y and capital R. So, but gametes are formed 4, but are not different type of gametes. All gametes are similar. So, but here it is heterozygous condition. We are getting 4 different type of gametes. So that's why whenever you ask, for example, given a genotype, write the possible number of gametes. Write the possible number of gametes. You always choose heterozygous condition, and apply the formula 2 to the power of n. So by this dihybridization, Mendel proposed which type? What is the law? Law of independent assertment. Law of independent assertment. So this is the Mendel's second law. Mendel's law of independent assertment states that allele pairs separate independently during the formation of gametes. This means the traits are transmitted to offsprings independently one another. That means the pair may not fixed. Round and yellow always fixed. It is not fixed. Round and yellow are each character is free from other. Each character as independent, which over the other. So that's why at the time of gametogenesis, at the time of meiosis, the round and yellow, both characters are separated. They randomly united, and they form different combinations also. They form different combinations also. This law
Sir,
is yes.
can you repeat the formula that how to apply once again, sir?
This is 2 to the power of n. This is the formula. So n is equal to number of allele. Number of allele. For example, I will give 5 allele genotype. 5 allele genotype. In the 5 allele genotype, how many gametes are formed? You apply 2 to the power of n formula. 2 to the power of 5, you are getting some value. But I am asking about what are the possible different gametes? What are the possible different gametes obtained from this genotype? At that time, you choose how many are heterozygous condition. How many are heterozygous condition? Among 5, 3 are heterozygous condition. Just you can apply 2 to the power 2 cube. That is 8 gametes are different gametes are formed. Remaining gametes are similar. So whether how many alleles are in heterozygous condition, how many alleles are in homozygous condition, you always choose heterozygous condition for getting different gametes. For getting different gametes. If it is all are homozygous, the number of gametes are formed is similar, same, but similar type of gametes are formed. That is the you have to remember when you apply this formula 2 to the power of n. Are you followed?
Sir, how we will get to know that 3 are the heterozygous condition?
You know, that is a capital Y is there beside a small y is there, no?
Yes, sir. Yes, sir.
That is heterozygous condition. That's all. So this is
Okay.
heterozygous condition. So this is heterozygous condition here. If it is
Okay.
2 capital letters are there, that is homozygous.
Homozygous.
2 small letters are there, homozygous condition.
Yes, I got it, sir.
Okay. That's you can choose some numerical problems asked in the genetics portion. That's why I'm stress on this part. Okay. So the second law, the law of independent assertment, a pair of characters may not fixed. It is free from each other, and it is randomly united and form different combinations. That's why we are getting round and green and wrinkled and yellow, and different combinations are formed in the dihybrid ratio. Out of 16, how many parental characters are found in the dihybrid experiments? Out of 16 progeny, number of plants shows parental characters. I am asking. Before that, what is the phenotypic ratio of the dihybrid?
9:3:3:1.
Yes, total 16 plants are there.
Yes, sir.
Among 16, how many plants with parental characters?
9.
Wrong. 10.
4, no?
9:1 is a
Oh.
parental combination. How many plants shows non-parental combination?
6.
8.
Yes, 6. 9 and 1 is parental. Middle 3 is non-parental. Yes or no?
Yes, sir.
That is very important. How many plants are shows the parental combination in the F2 generation? So 10 plants are shows parental combination. Non-parental combination. That indicates what? Non-parental combinations are coming in the Mendelian experiment that shows law of independent of assortment. Law of independent of assortment. So this indicates that the pair of characters are not fixed, which are free from each other and freely segregated and randomly united and form different combinations that shows in dihybrid experiments. Is it clear?
Yes, sir.
Okay. So this is about Mendelian inheritance laws. So this is, you know very well. Parental combinations always homozygous. See here, capital R, small capital Y, and here small r, small y, small y. So this is the parental characters. Out of 16, this you can see here. Resulting genotypes 9/16, which containing capital R gene and capital Y gene, it indicates the parental character. So, and one is purely homozygous, that is recessive genes, and the remaining 3+3 is non-parental combination. So, resulting phenotypes are. What is the genotypic ratio of the dihybrid?
1:2:1, 2:4:2, 1:2:1.
If you got confusion, just you remember like this: 4 2s, 4 1s, 4. What is
Yes,
that?
sir.
4 2s, 4 1s, 4. Yes, in the ratio lo emuntayi 2 అనేటువంటి digit 4 times repeat అవుతుంది. 1 అనే digit 4 times repeat అవుతుంది.
4 1.
4 2s, 4 1s, 4. Are you clear?
Yes, sir.
So this is the cell for 9:3:3:1 ratio is the phenotypic ratio, and the genotypic ratio is there. So these are the characteristic ratios are very important. Normally asked in examination. Everybody knows 3:1, 1:2:1 is the monohybrid ratios, and 9:3:3:1, and 4 2s and 4 1s and 4, that is the dihybrid ratio. We can see with a number of examples. Is it clear? So this is the genotypic ratio here. So 1:2:1, any ratio we can write in a different form, but our ultimate is 16 are present or not, that you can choose here. Two pure plants of a species in order to study the inheritance of 2 pairs. Trihybrid and 3 pairs of size. So atla man different type of ratios we can easily identify. So today we'll discuss about some terminology and Mendel experimental tool plant details, and why Mendel choose pea plant for his experiments, and what is the secrecy of the in the inheritance, what type of inheritance laws are proposed by 2 experiments, that is monohybrid and dihybrid, and these Mendelian genetics is applicable to only nucleus. It is not relevant to the plastids, or not relevant to the mitochondria, not relevant to the plasmid. Because plasmid, mitochondria, plastids are which are present in where? Cytoplasm, which are present in cytoplasm. But Mendel always focused on the genes which are present on the chromosomes. Chromosomes are completely embedded in the nucleus. So that's why Mendelian genetic is applicable to nucleus, not for the extranuclear genome. Not for the Mendel laws are not applicable to extranuclear genome. Extranuclear genome means some amount of DNA which is present in cytoplasm also, other than the nucleus. Other than the nucleus means mitochondrial DNA is there, chloroplast DNA is there, plasmid is there. So plasmid and mitochondrial DNA and chloroplast DNA, it is not relevant to the Mendel laws. It completely against to the Mendel laws. Is it clear? So in the tomorrow class, we can discuss back cross, test cross, reciprocal cross, different type of process in the genetics. Are you followed this part? Yes, sir. Okay.
Really, it's a wonderful class, sir. The genetics, Mendel laws,
Okay.
and ratios of the Mendel laws, how ratios are occurred, and how to remembering of this laws of the ratio of the different laws of the Mendelian, and also some of the terminology related to our the genetics. Clearly, you are delivered in this class, sir. Really, our participants, they are get benefited. Majority of they are involved during your class. Whenever you are deliver your lecture, all the participants, they are enthusiastically involved your teaching learning processes. Really, it is a live class, sir. Really appreciable class. And also, thank you very much for your more than one and a half hour. You are sparing your valuable time here. So tomorrow also same time, please your valuable time for our young aspirants. And also it is useful for JL, DL and other competitive exams of our plant sciences. It is most important area.
Okay.
So really, thank you.
Thank you, sir. Thank you very much for your patient listening. Okay, sir. I'll leave it.
Yes, sir. Thank you. Tomorrow we will meet.
Okay. Okay.
So next class sharp 2 o'clock. Next class 2 o'clock.